Gene expression and regulation. Findings. We have here the number 1. Genes can't control an organism on their own. Rather, they must interact with and respond to the organism's environment. Some genes are constitutive or always on regardless of environmental conditions. Number 2. Such genes are among the most important elements of a cell's genome, and they control the ability of DNA to replicate, express itself, and repair itself. And lastly, these genes also control protein synthesis and much of an organism's central metabolisms. In contrast, regulated genes are needed only occasionally. But how do these genes get turned on and off? What specific molecules control when they are expressed? It turns out that the regulation of such genes differs between prokaryotes and eukaryotes. For prokaryotes, most regulatory proteins are negative and therefore turn genes on or off. Some repressors bind near the start of mRNA productions for an entire operon, or cluster of co-regulated genes. Others have a fine-tuning system which uses mRNA structures to stop both transcriptions and translations, depending on concentrations of an operons and product enzymes. And yet another layer of regulations affects the structure of RNA polymerase, which turns on large group of genes. While for eukaryotes, default state of gene expression is off rather than on. As in prokaryotes, the secret lies in chromatin, or the complex of DNA and histone proteins found within the cellular nucleus. This code includes modifications of histones, positively charged amino acids to create some domains in which DNA is more open and other tightly bound up. It is possible for cells to maintain the same histones code through many cell divisions. For transcriptions to occur, the area around a prospective transcription zones needs to be unwound. This is a complex process requiring the coordination of histone modifications, transcription factors binding, and other chromatin remodeling activities. Each TF has a specific DNA binding domain that recognizes a 6 to 10 base pair motif in the DNA. In the test tube, scientists can find a footprint of a TF if that protein binds to its matching motif in a piece of DNA. For an activating TF, the effector domain recruits RNA polymerase II, the eukaryotic mRNA-producing polymerase, to begin transcriptions of the corresponding gene. Some activating TFs even turn on multiple genes at once. All TFs bind at the promoters just upstream of eukaryotic genes, similar to bacterial regulatory proteins. However, they also bind at a region called enhancer, which can be oriented forward or backwards and located upstream or downstream or even in the introns of a gene, and still activate gene expressions. Because many genes are co-regulated, studying gene expression across the whole genome via microarrays or massively parallel sequencing allows investigators to see which groups of genes are co-regulated during the differentiations, cancers, and other states and processes. Most eukaryotes also make use of small non-coding RNAs to regulate gene expression. For example, the enzyme dicers find double-strand regions of RNA and cut out short pieces that can serve in regulatory role. Argonaute is another enzyme that is important in regulations of small non-coding RNA-dependent system. Imprinting. Imprinting is yet another process involved in eukaryotic gene regulations. This process involves the silencing of one of two alleles of a gene for a cell's entire lifespan. Imprinting affects a minority of genes, but several important growth regulators are included. For some genes, the maternal copy is always silenced, while for different genes, the paternal copy is always silenced. The epigenetic mark placed on these genes during egg or sperm formations are faithfully copied into each subsequent cells, thereby affecting these genes throughout the life of the organism. Still, another mechanism that causes some genes to be silenced for an organism's entire lifetime is X inactivations. In female mammals, for instance, one of the two copies of the X chromosome is shut off and compacted greatly. This shut-off process requires transcriptions, the participations of two non-coding RNAs, one of which codes the inactive X chromosomes, and the participations of a DNA-binding protein called CTCF. As the possible role of regulatory non-coding RNAs in this process is investigated, more information regarding X inactivations will not be discovered.